those of you who are joining via the web, we're just waiting a few more minutes for additional people to arrive.
How many will join online?
Yeah. I think we can start whenever, because actually of all the people who registered, almost all of them are on Zoom.
Oh, okay.
Okay, great. Well, so it's my pleasure to introduce our speaker for today. I'm sorry. Pennell, she came all the way from Charleston yesterday. So today, for Dr. DeLeon Pennell, she got her bachelor's degree at Baylor University in 2007 in biochemistry. She stayed to continue her graduate education there, obtaining her PhD in biomedical studies in 2011. From there, she did a few postdocs and eventually sort of moving east, and eventually in San Antonio and in Mississippi, eventually landing in Charleston in 2018 as assistant professor at MUSC. So today, Dr. DeLeon Pennell will talk about cytotoxic T cells. So we're certainly happy to have you and look forward to your talk.
Thank you. So I'm very happy to be here to talk a little bit about my research that I've been doing since I joined MUSC in Charleston. And so we're going to talk a little bit about the heart and cardiovascular disease. So obviously, cardiovascular disease is the #1 killer in the United States. And so we know that, especially here in the South, cardiovascular disease prevalence is significantly elevated. And we know that this is the #1 killer in the United States. And so obviously this is a significant pathology in not only in the United States, but in the world. And so in my lab, we mainly focus on a myocardial infarction or a heart attack. And so we know that approximately every 40 seconds, an American is going to have a heart attack. And this is caused by an occlusion of the blood supply to the myocardium or to the heart. This causes a necrotic response. So all of the heart cells are there within that region that are no longer getting blood begin to die. This is going to cause inflammatory response and thus initiating the whole wound healing response. And so we know this is a disease that tends to affect the older population. So men tend to have their first heart attack around 65 years old, where women don't have it until around 72 years old of age. And so my lab mainly like to look at the immune cells and what are the immune cells doing in regulating this whole inflammatory response, remodeling, and initiating that scar formation. And lately, in the last couple of years, we're starting to look at the cytotoxic or CD8 positive T cells. And so in the literature, CD8 positive T cells are mainly known for their role in killing other cells. This is mainly in cells that are infected with viruses and bacteria, and also cancer cells. And so they have three main known functions. They secrete cytokines such as TNF-alpha and interferon gamma. These will induce apoptosis and the inflammatory response. They also express fast ligands. These are co-stimulatory molecules that activate pathway signaling in the target cell. And they also secrete cytotoxic granules such as granzymes and perforin. This will actually induce lysis of those target cells. And so what we do know post-MI is that there's an influx of these CD8 positive T cells, and in the permanent mouse occlusion model, Leon et al. Found that at 1 day after occlusion, you start having this infiltration of these CD8 positive T cells into the infarct. They start peaking around day 5, day 7, and have been found to be within the infarct as long as 8 weeks after the initial injury. So we know they're there in the infarct, but we really don't know what's going on. What is their job? What are they actually doing? And what is their role in the whole infarct remodeling and healing process? And so in clinical research, we also have found that granzyme B, which is one of the main granzyme proteins that are released by CD8 positive T cells, have been correlated with an increased risk of death in post-MI patients at 1 year follow-up. And so in this recent paper by Cytotoxic that just came out earlier this year in 2021, they found that elevated levels of this granzyme B protein in the plasma were actually could predict cardiovascular death in these post-MI patients. And so, as I mentioned, we really don't know what they're doing in the post-MI heart. So there's really only been about 3 different studies looking at CD8+ T cells and post-MI remodeling. And so one of those studies, they actually looked at CD8 T cells. They induced an infarction, isolated the CD8 T cells from the infarct, and co-cultured them with either myocytes or what they considered non-myocytes, which was mainly a macrophage population. And they looked at what happened to both the T cells and to the other cells when they co-cultured the control healthy T or the CD8 T cells from a control healthy heart, a sham heart, or the post-MI heart. And as you can see from this graph, let me get my laser. So you can see from this graph, the lymphocytes, when cultured with myocytes, all had more proliferation than when they compared to those non-myocytes, no matter whether it was sham or the MI CD8+ T cells. In addition, when they looked at the myocyte destruction or the cell death, they found again that when they took CD8+ T cells from the MI heart, there was more cell death in those myocytes. And so, like I said, CD8+ T cells are known for their role in causing death. So from the study, they hypothesized that the CD8+ T cells were antigen specific and that they were initiating myocyte death due to the initial infarct injury. And so we think that it's probably a little more complicated than just initiating myocyte death. And so we have this genetic mouse model of CD8 T cell depletion. So the CD8 receptor is necessary for CD8 T cell activation. This receptor has to be present on this T cell to be able for it to become activated. And so in this model, we have a mutation on the CD8 receptor. This causes the receptor not to be trafficked to its cellular surface. And if it's not expressed by the cell on the cellular surface, then it cannot be activated. And so in this mouse model, there is actually very little changes to the CD4 population. So T cells can be classified by their CD8 or CD4 expression. And so because we like this depletion model, because the CD4 in a healthy animal is not affected. And so we wanted to look using this animal model at what happens after an MI. So first we used this animal model and we performed a permanent occlusion surgery on these animals, and we looked at survival. And so we've taken this out to day 14, but in this graph you'll see this is to day 7 post-MI. And so what you'll notice is at day 7, about 60% of our wild type animals will live after the occlusion. And so that's very typical. We do have a lot of animals that die from the remodeling, whether it's due to heart failure or sometimes we have ischemic rupture, so the hearts tend to rupture. What was interesting, however, was our CD8 T cell deficient animals had significant improvement in their survival. So we had about 75% of these animals actually live to day 7 post-MI. And one thing to know about the study is we really wanted to look at the remodeling process. So how well does that scar form? So we limited our data set so that the infarct sizes were the same. So this has nothing to do with the injury size and everything to do with how well does the scar form. And so what you can see here, this is what we call TTC staining. So our hearts get incubated in this chemical called TTC. It will stain the viable tissue red, which you see here. This is what we call the remote zone. So this is the tissue that is still alive, and everything that is not alive is white. So you can see that here. That's our infarct zone. And what you also can see from these two different sections of the heart, so this is the midsection of the heart, is that just looking at it, you can already tell that there's a significant decrease in LV dilation, which we then confirmed doing echocardiography. Sorry. So echocardiography of our animals. So this is looking at cardiac function. How well does our animals pump blood after that initial insult? So this is at day 14 post-MI. And so Miguel Troncoso is my surgical guy. And so he actually does both my surgeries and a lot of my echoes for my lab. And he found that 14 days after the initial insult, there was a significant improvement in what we call ejection fraction. So that is the amount of blood that is being pumped out of a heart. So normal ejection fraction for a mouse, and a human actually, is going to be around 65 to 70%. So 65 to 70% of your blood that comes into the heart will eventually get pumped out of the heart. And so you see at 14 days post-MI and a permanent occlusion animal that there's a significant decrease in the amount of ejection fraction, and this was improved in our CD8 T cell deficient animals. You also can tell that the end-diastolic volume, so this is how big that heart got during diastole, so relaxation, and the systolic volume, which is during systole, so this is during contraction, was significantly decreased in our CD8 T cell deficient animals. So not only do we have an improved ejection fraction, we have a decrease in that LV dilation. So we wanted to know, okay, what is the mechanism here? So what is actually happening? What are the CD8 T cells or CD8 T cell depleted animals? What is happening to causing this improvement in survival and cardiac function? And so I had two master's students that were in my lab last year, and they basically hit the ground running, and for 3 months, all they did was histology. And they staged our hearts for macrophages. And so we know that macrophages can come in as early as 24 hours after the occlusion, peaking around day 7 and then going away by day 14. And you can see this here in the wild type animals. And so you see macrophages beginning to infiltrate, starting to increase at day 3, peaking around day 7, and going back down by day 14. What was interesting, however, was in our CD8 T cell deficient animals, you have this delay. So there's almost no macrophages infiltrating at day 1 post-MI, there's very few. Then all of a sudden you have this drastic increase. This is almost double what we normally see in a wild type animal. Day 7, no significant difference between the two genotypes. And then at day 14, it remains slightly elevated compared to the wild type. So it's almost as if there's a delay in that macrophage recruitment. There's a shift to the right, and as far as the timeline is concerned. And so we wanted to know, well, why are the macrophages not coming in? And is it, if you have a beneficial response and we have this delayed inflammatory recruitment, is it possible that there's actually a difference in the type of macrophages that are coming in? Yeah.
How many animals in each group?
So this is at least an N of 3, because I think the day 14 was a small sample size, but all the other ones were at an N of 6 to 8. So it's a fairly large sample size for most
Thanks
of
very much.
So we know that macrophages do not come in one size, one type. So we know that in a normal heart, there's resident cells. These resident macrophages, their job is to basically go around the heart, making sure that if there's any viral infection, any type of injury, they're going to respond to that, and they're going to initiate the healing response. So we consider the resident cells to be beneficial. After infarction, however, the resident cells are totally killed within that infarct. Because of the ischemic insult, they do undergo necrosis, and then we have to basically replace them with our bone marrow cells. So our bone marrow begins to make more monocytes, these infiltrate into the infarct, and these resident cells are replaced by bone marrow monocytes. These bone marrow monocytes are not typically beneficial long term. They're good, we need a little bit because we need to remove all that dead tissue, but long term, we don't think they're actually beneficial for the whole wound healing response. And so with all the single cell data sets that we have out there, there's one paper by Dick et al. That came out in 2019, and they looked at a day 0 heart and day 11 post-MI heart, and they clustered these macrophages and found that there was about 11 different clusters in the post-MI heart. Interestingly, the top 6 clusters were similar to what we would consider a resident cell. They did not express some of the main markers that we would consider a resident marker, but many of the other markers were very similar. So we considered these are resident-like monocyte-derived macrophages. And so what we wanted to know is, because we have this shift in the macrophage timeline, do we also have a shift in the macrophage phenotype that are coming in? And so using this data set as a guide, we have this RNA bulk seq data that we had generated looking at day 1, 7, and 14 post-MI. And I had a medical student, Roberto Muniz, who basically took our 15,000 genes, pulled the ones that were identified in all these different clusters, and then clustered our data set to say, do we have differences in resident-like populations within our data at these different time points? And so doing that, we then looked at those different clusters over the time point post-MI and found that a couple of them that were known to be resident-like populations, specifically cluster 1, 4, 5, and 6, were actually elevated at day 7 and 14 in our CD8 T cell deficient animals. And so you can see that these are all the genes associated with that cluster that were within our RNA bulk seq data set. And we normalized, log transformed these so that they could so that not one gene would then influence the whole cluster. And then we looked at the statistical difference between those two groups. And so as you see, the day 7 sets, other than actually cluster 4 is actually lower at day 1, but higher at day 7, there was a significant upregulation of these resident-like markers in our NFARCs of the CD8 T cell deficient animals. When we looked at the other clusters, so clusters 7, 10, and 11, so these are the ones that don't seem to represent those resident-like macrophages. We also saw a couple other differences. So cluster 7 is a CD209A dendritic cell cluster. This was also elevated in our CD8 T cell deficient animal model. And cluster 10, which when we put into gene ontology, seems to represent a cluster that may be regulating endothelium. And cluster 11, which we think is regulating exocytosis, were actually also elevated, but this was at day 14. And so there seems to be some specific cellular population changes that are associated with this change in the timeline of macrophage recruitment. And so one thing to consider is we really didn't see any day 1 differences, but this may be a caveat of the big data set that we use to cluster our data, right? Because the data that we used was from day 11 post-MI animals, which we know post-MI there's a temporal change. The initial influx of monocytes are going to be all pro-inflammatory. And as that wound begins to heal, you have a change in the way those macrophages behave. And their phenotype becomes more anti-inflammatory, pro-healing. And so by looking at day 11, when we're in that pro-healing, pro-resolution phase, it may not actually reflect what's happening at day 1. And so what we want to do now is look, because we didn't see many differences at day 1, using this data set to cluster our data, we want to go back and look at some of the other single cell data sets that are out there and looking at the day 1 clusters. Can we see differences there based on some of the earlier, more acute time points? Because this may not actually reflect what's happening at day 1, since they use the day 11 data. So, but that still leads us to multiple new questions, right? So we have this altering of the macrophage timeline. These macrophages are coming in later. They seem to be more resident-like, which would be beneficial for healing, which would explain why we have better survival, decreased dilation, improved cardiac function. So what is the stimulus that is causing these macrophages to change the recruitment, to change the phenotype? And so there's 3 different possible mechanisms. The chemotactic signaling, so obviously after an injury, you're gonna have all these different chemotactic proteins that are gonna be secreted into the circulation. These are gonna initiate that response. All the monocytes are gonna be recruited to the infarct. So is there something different there? The other one is the MI microenvironment. So this is a newer field where where people are starting to look at within the infarct, are there differences in the lipid profile? Is this what's causing differences in how well those immune cells are being recruited? And the last one is spleen recruitment. And so we know the spleen is a reservoir for monocytes, so it's not just bone marrow, but it's also the spleen. And is there a difference in how well the spleen is being tapped into after the MI with the CD8 T cell deficient animals? And so we wanted to look at those 3 different areas. And so first we looked at what is known in the literature as far as CD8 T cell regulation of monocytes. And so in atherosclerosis and thrombosis, we know that there's multiple chemokines that can be secreted that can regulate monocyte recruitment. CCL2, CCL5, CXCL1, interferon gamma, BCAM are all a couple of chemokines that have been associated with CD8 T cell regulation of monocytes. And so we did a plasma proteomic profile. So we use this, what's called the mouse XL cytokine array. It's by R&D Systems. It looks at over 100 different proteins. And doing this, we looked at day 3 and day 7 specifically in the plasma. And we tried to determine, is there a specific cytokine within our animals that may be explaining why we see this delayed recruitment or delay or altered activation status in our monocytes. And what's interesting is we really didn't see very many differences at day 3, and you can see this cluster data. So this is the average of our N, so this is N of 6. And at day 3, here's our wild type, here's the CD8 T cell deficient animals, and you can see really very little differences overall in those different chemokines. When you look at day 7, however, this is where we started seeing our differences. So the proteins that are associated with leukocyte chemotaxis really didn't have a drastic difference. However, some of them that were associated with MAPK kinase or K signaling and angiogenesis were actually upregulated in our CD8 T cell deficient animals, where they were downregulated in our wild type. And so one thing that we do know post-MI is the plasma doesn't always reflect what's happening in the heart. Because in the plasma, we have this robust increase of all this inflammation very early. And by day 7, we see a lot of decrease in the chemokines. By day 7, we see very little chemotactic proteins in the circulation. So it may be that the heart may not quite reflect what's going on. So we then started looking at the heart specifically and doing a different ELISAs at the specific cytokines that are associated with CD8 T cell regulation of monocytes, and we didn't see very much difference. So interferon gamma did not significantly increase. There was no differences between the groups. CXCL1 does go up early post-MI in both genotypes, but there was no difference again between those groups. And so it doesn't seem like there's a difference in the chemotactic signals when CD8 T cell deficient animals are lacking, which we were kind of surprised because we definitely expected to see something there. And it may be that we're not looking at the right chemokine, so there is that possibility, but as of right now, we're not seeing any chemokine differences. So we then went to our second hypothesis that there's something different within the MI microenvironment. And so a lot of the more recent literature started looking at this lipid metabolism, spinosyn, prostaglandin, what is happening, how is that affecting the whole recruitment of immune cells? And so we looked back at our RNA bulk-seq data set and looked specifically at those genes that are associated with regulating lipid metabolites. And so what we did notice, so everything that's shown in green are those that are downregulated in our CD8 T cell deficient animals, and everything that's shown in red is upregulated in our CD8 T cell deficient animals. And so we do have a couple of signs that there may be some differences there. So the spinosyn kinase is actually downregulated, MMP9 is downregulated, a couple of the prostaglandin synthases are also downregulated. But what's also interesting is this S1PR5. So this is a receptor that is expressed by monocytes and actually will cause them to traffic into the inflammatory site. We did find it to be upregulated in our bulk RNA-seq data. So it's possible that there is some more alterations there within the lipid environment that is causing this immediate huge increase at day 3, specifically post-MI. And so this network, this is at day 14 post-MI. So this is a reflection of what is happening at day 14. So we also did some single cell recently, and so I don't have a lot of single cell to show you. However, what was really, really interesting to us is when we compared our single cell to our bulk RNA-seq, some of those genes that were found to be downregulated in our CD8 T cell deficient animals were actually identified in cluster 5 of our single cell. So it's very possible that the reason why they were downregulated is that the CD8 T cells are actually a source of these different kinases and synthases, and that they may actually be facilitating in this alteration of the lipid environment. And so that's something that we're starting to kind of look at. We haven't gone into a lot of details in quite yet, but we are really interested in, we know that the lipid environment can affect CD8 T cells, but can they also do the opposite? Can they then in turn regulate the lipid environment? And so that's something that we're really interested in now. And so the last mechanism that we really wanted to test was this whole idea of the splenic reserve being important for monocyte recruitment early post-MI. And so in a study by Dvorsky and Nachendorf back in 2009, they found that within the first 24 hours, the spleen is actually where the majority of the monocytes come from. So it's not the bone marrow initially, it's actually the spleen. And the spleen acts as this reservoir for monocytes during that first initial ischemic response. And so they found that during that first 24 hours, the spleen is where they're all being recruited. And they took this a step further and started looking at what is that signal. And they interestingly found that the angiotensin is actually one of the signals that causes these splenic monocytes to leave and infiltrate or come into the circulation to eventually infiltrate into the infarct. And so they found in this study where they used the angiotensin converting enzyme inhibitor that they could actually decrease the amount of splenic monocytes from infiltrating into the infarction. So we wanted to know, okay, do we have any differences in the way the spleen is releasing its monocytes? And so I have another technician. So Philip actually came here. He was a student here at USC. And he's actually about to go to medical school, which we're very happy and proud of him for. He looked at the spleen, and so we looked at specifically at day 3, which we thought by day 3, the spleen should be totally emptied of any monocytes if it has been tapped into. And so we looked at the spleen at day 3 post-MI by flow cytometry, and we did find that there was more monocytes within the spleen in our CD8 T cell deficient animals compared to our wild type animals. So we do think that the spleen, for whatever reason, is not being tapped into. And so the spleen is not releasing monocytes, and that would explain why at day 1 post-MI, we don't have the initial recruitment into the infarction. And because we know that the angiotensin pathway is one of the signaling pathways to initiate that recruitment, we then went back to our RNA-seq data, looking specifically at those different proteins, and the only one we found at day 1 post-MI that was different is actually angiotensin converting enzyme 2. So as you can see, wild type CD8 T cell deficient animals was actually significantly upregulated at day 1 post-MI. And so we know that angiotensin, ACE2 is actually important for converting angiotensin 2 to that ACE1-7 peptide. And we also know that it's through the AT1 receptor that the monocytes are actually being activated to leave the spleen. And so because if this is upregulated and we have this conversion from angiotensin 2 to 1-7, now we have a activation of MUSC, which is one of the receptors that 1-7 binds to. This will cause a activation of vasodilation, vasoprotection, and inhibit tissue damage and edema. Where if we had more angiotensin 2, we would obviously have vasoconstriction, inflammation, fibrosis, and more tissue damage. So we think something about this elevation of ACE2 in our CD8 T cell deficient animals is actually what's in keeping those monocytes from leaving the spleen. And so that's the signal that is inhibiting our splenic reserve from being tapped into.
You have a question from
Okay.
the audience. I don't know if you prefer to keep them till the end or to answer them as they come. So if I try to read it off, I'm gonna embarrass myself. So you
Okay.
can.
Is ACE2 is increased, I would guess increase in bradykinin as well. So that's a great question. We have not looked at bradykinin. We did try to do a couple measurements in the plasma for angiotensin 2, and we haven't been able to find a good assay to measure that. So if you have any suggestions on what the best way to measure either bradykinin or angiotensin 2, we would be very, very grateful. But yeah, so all the different ELISAs that we looked at, and I asked a couple of different people who are, more in that field, and they said, yeah, they're not very clean. So any suggestions, we would love that, because we actually have not looked at bradykinin, and I don't remember seeing in our RNA-seq data bradykinin being different, but that is a good question. And then there's another one, was any difference in COX expression in CD8 T cell knockout mice? Another great question. I'd have to go back to the RNA-seq data. I do not remember on top of my head if there was. All right. So, as I mentioned already, so we know that wound healing is spatial and temporal dependent. So the MI wound is not static. It changes as the wound begins to heal. So we have the inflammatory component. That's where it's very highly pro-inflammatory. That's where the necrosis happens. You obviously have this huge infiltration of neutrophils, macrophages, T cells. They're going to come in to remove that necrotic debris. So all those dead myocytes and other cells that have died due to the ischemia. This will then, once all that necrotic tissue has been removed, you start having that proliferation response. This is where fibroblasts begin to infiltrate, they become activated, and they start to start laying down that collagen. And then you have your maturation. This is really where the scar is beginning to mature. The collagen is not only being laid down, it's being cross-linked, and you have that scar formation and fibrosis. And so we know that all these alterations to the inflammatory response is obviously going to change the way the scar is formed. And so we wanted to know now, if there's all these changes in the macrophage timeline in the phenotype, what does this do to the scar? And so I had a undergrad, Kimberly Oviedo, who was there over the summer. And then I also have a current graduate student, Alexa Corker, who is now an expert at the confocal. And so they both started looking at a little bit more at fibrosis and different components of that. And so Kimberly actually did what we call picoseries spread, looking at collagen in our infarcts. And looking at the time course post-MI, we know that over time, we start having a huge scar develop in our infarcts. And you can see this by this red stain here, that is all the collagen that is being laid down within that infarct. And so what was interesting, however, was in our CD2 deficient animals, we do start seeing collagen being laid down a little bit earlier. So at day 3, we see collagen being laid down. That's obviously where those macrophages all of a sudden begin to peak and come in at high numbers. At day 7, there was no difference, but then by day 28, we do start seeing a possibility of it's going to continue to increase. And just looking at the picoseries red stain, you can actually see that the fibrils almost look thicker. So we are actually starting to look at those fibrils a little bit more in detail. What is the characterization of those fibrils? But it does seem like they may be actually thicker as in our CD8 cell deficient animals. We then looked at a marker of fibroblast activation. This is alpha-synuclein, and this was the green stain here. Blue is obviously DAPI, so that's a nuclei. And we did find that in our CD8 cell deficient animals, there is actually more of this alpha-synuclein. So the fibroblasts seem to be more active, thus they're making more collagen and they're making more ECM for that scar. And so it does seem that there is more ECM within that scar. This would obviously, we would indicate that maybe it makes it stiffer. But obviously collagen is not all the same. Collagen cannot just be laid down. It has to be formed into those different fibrils. This is going to be done by different enzymes that will cross-link. And these different enzymes, including lysyl oxidase, so lysyl oxidase like 2 that was shown in this paper, are going to facilitate in the way that collagen is formed. And so in this paper, they looked at liver fibrosis, and they found that inhibiting lysyl oxidase like 2 would actually make a way for macrophages to infiltrate. So remember, we have this huge infiltration of these macrophages at day 3 post-MI. So we wanted to know, is it because we have changes in the lysyl oxidase and thus changes in the way that collagen is being cross-linked? And so we looked at our data set so we wanted to know, is there decreased cross-linking? And is this why we have that huge infiltration of monocytes at day 3 post-MI? And so going back to, so we went to our RNA-seq data for pretty much answering all the questions. And so we went back to our RNA-seq data, found again, there wasn't a huge difference in most of the enzymes that are associated with cross-linking and formation of the collagen fibrils. There was a couple, lysyl oxidase 3, a couple different hydroxylases but really not much differences. So I'm going to highlight a couple. So lysyl oxidase here, that's shown here by the arrow. This one is actually an HIF1 alpha marker. These were actually also identified in our single cell data set. So it does seem that when we do know, based on other people's data, that T cells can make cross-linking enzymes. And so it's possible that even though it wasn't identified in our single cell data or in our RNA-seq data, that there may be some changes in cross-linking just due to the lack of CD8 T cells in the infarct. And we also measured protein levels, and so we did find that the active lysyl oxidase form does seem to be slightly decreased in our CD8 T cell deficient animals. So if we do have decreased cross-linking of this collagen, it doesn't matter how much is being laid down, if it's not being cross-linked, then that may be actually what's causing this huge infiltration of macrophages into the infarction, because there's more space for them to squeeze because that collagen isn't super tight. We also looked at other components of collagen. So we know that collagen is being laid down, it gets cross-linked, but you also have MMPs, so matrix metalloproteinases, bone matrix proteins, atoms. All of these proteins are actually doing the opposite. So they're degrading collagen, right? So there's this constant balance of keep regulating the extracellular matrix, and that's what causes a wound to be what we consider a good wound or a bad wound. And so we then looked at all the different MMPs, BMPs, atom TSs within our data set and clustering them all together, looked at is there any differences over the time course. And interestingly enough, there was actually more overall BMPs and MMPs, proteases in general at day 1 in our CD8 cell deficient animals. No significant difference really at day 7. There was actually a significant slight decrease, very slight, and it remained elevated at day 14. So almost like the CD8 T cell had this elevated and constant protease activity, where the wild type had an up-down profile. And when we look specifically at what MMPs this may be, contributing to this, what we did find is MP9 was drastically lower. And so MP9 is a huge player in the post-MI remodeling process. It has been linked to development of heart failure, which is what we're really trying to limit, is development of heart failure after ischemia. And it is almost 100-fold lower in our CD8 T cell division samples. We also found lower levels of ADAM-TSs, 1, 4, and ADAM-8. We did find a slight decrease in MMP2 at day 7 post-MI. However, ADAM-TSL2, TS2, ADAM-TS10, and BMP1 were all elevated at either day 1 or day 14 post-MI. So there does seem to be somewhat of a dysregulation as far as what MMPs are up and down. But the most interesting data to us was the fact that this MMP9, which is a known and huge player in remodeling, was so drastically decreased.
Did you see similar levels of decrease in MMP9 activity?
So we haven't done like a zymogram or anything. We did do a Western blot looking specifically at pro and active bands. And we did not look at day 14, which is where we see that huge drastic decrease. So we haven't looked at day 14 yet. But at day 7, we do not see as much in the active band. It's not as drastically decreased. So we really need to look at day 14, which is where we see this huge genomic component that's decreasing, but we haven't done that quite yet. We also looked at TIMPs. So TIMPs are the tissue inhibitor protease, metalloproteinase inhibitors. So they inhibit MMPs. And so we looked at how they changed over the post-MI time course. There was decreases in TIMP1 in our CD8 cell division animals at day 1 and day 14, where TIMP2 and TIMP3 were actually elevated at day 1 and day 14, and no differences were in TIMP4. So again, there seems to be definitely a dysregulation of how these MMPs and the protease activity of this overall protease activity in our CD8 cell division animals. And we do know that CD8 cells are a source of proteases. So it's possible that that may be contributing, because if they're not there to secrete their MMPs, thus they're not going to actually be able to degrade collagen. So to kind of summarize what we know so far, and so we're still trying to digest all of this data that we've generated within, really majority of what I've showed you has actually been within the last year. So we definitely think that the CD8 T cells are really important, not only for killing myocytes. So I hope I've shown you that at least. So they're not just there just to kill the myocytes and just to initiate infarct expansion, but they also are regulating that initiation, so the recruitment of those monocytes. And so we do think that there's both an angiotensin component as well as regulation of this lipid microenvironment, and that may be a role of the CD8 positive T cells post-MI. And we also have some evidence to suggest that they are really important for maturation of that scar, and whether that's through direct release of lysyl oxidases or indirect through regulation of the inflammation component. But we do have some evidence to suggest that the scar is definitely going to be a lot stiffer in our CD8 cell deficient animals, and when we give CD8 T cells back, the stiffness is drastically decreased. And so there definitely seems to be some type of regulation process within the maturation component, and CD8 T cells are definitely playing a role. And so right now we're really trying to tease out what all those different components are doing, but obviously the biggest thing for us is trying to find balance, because we know inflammation is necessary. We have to have it, but we don't want to get rid of inflammation. But we do need to find that balance. We don't want them to have chronic inflammatory status, because this is when we get into the pathological conditions like heart failure. We want them to have that initial influx of inflammation so that the necrotic debris can be removed, the scar can be developed, but then we need them to go back down to some type of new homeostatic state. And that is actually what we're trying to do, is find out how do we tweak it so that we don't totally ablate inflammation, but tweak it in a manner so that they can clean up the debris, but still have a good scar. And so I just thank y'all for inviting me, and then obviously thanks to my lab and all the hard work they've done on this.
So much for a very nice talk. Are there any questions?
I think there's a, maybe we can answer the question online.
Oh,
first,
sure.
Yeah, so there's one. So TIM 3 regulates Atom 17 activity. Did you check Atom 17 levels? So Atom 17 was definitely in our ref seq, and it was not significantly different between the days 1, 7, and 14 that we looked at. So there was no difference as far as the gene level is concerned between our two genotypes.
Really nice talk, really enjoyed. I have two questions. First one is that you mentioned about the lipid distribution. So I missed that part. Did you check, what's the level of different kind of lipid?
So we have not. So, I mean, it really wasn't until our RNA-seq data that we started seeing those kinases and those syntheses and kind of giving us an idea that maybe there's some regulation there. And then we went to our single cell, we're like seeing kind of the same ones, and we're like, oh, maybe there's something here. So we haven't done that yet. This is something that I'm really wanting to do.
That'd be
Maybe
very
some lipidomics.
exciting, to see the distribution and different kind of, yeah.
Yeah, so we haven't done that yet.
Another question is a very general question here. You show a lot of data, for example, here, right? You mentioned, the spatial and temporal dependent, for the wound healing process is really important, right? But when you I would try to have to say, to analyze or somehow build up the connection between your mRNA results with your like say protein, right? And the plasma protein, the secretion, the level. I will try to do that,
So not yet. So majority of the time, if we find something that we think is really interesting, like the MMP activity, we're going to go then start looking at MMPs and look and measure a couple of them to look at the protein levels, activity levels. But we really haven't done that in a full scale yet. Like I said, a lot of this data we literally just generated in the last year. So it took us a while to kind of dissect it all and figure out all the different components that we are identifying what I showed. And so now we're really starting to look at what does this gene level change mean protein level.
Okay.
We've also have a couple collaborations with Dr. Zile and Dr. Amy Bradshaw, where we're looking at the fibrosis component and the biomechanics of the scar. And so it's not really looking at protein levels necessarily, but more functional levels.
Gotcha.
So how does this overall fibrotic change? What does that do to the scar stiffness? And so we're looking more at the functional differences, hoping to eventually kind of be able to link.
Yeah.
What is this gene level? What does that mean functionally? So we're kind of going more that way right now, but we definitely need to look at the protein levels for sure.
Thank you. I think there are more questions there.
Any suggestions about which prostaglandin or leukotriene that is involved in this response? That's a great question. Honestly, I'm not even sure. I mean, so we know that there are multiple prostaglandins and leukotrienes that are really important in the post-tumor remodeling process. We know that many of them, sphingosine 1, for example, has been shown to actually regulate inflammatory response. And we did do a Western blot specifically for sphingosine 1, and we found it was very dirty, but we did find a somewhat of a decrease in our CD8 cell deficient animals. But without doing a huge lipidomic profile panel or doing multiple Western blots, I'm not sure how we'll be able to dissect that out right now. So we're kind of we're trying to decide, do we want to go multiple Western blots or do a lipidomic profile? Figure lipidomic profile though.
Any other questions from the audience? I had a question about the clustering. What was the representation that you used? Have you? Oh, you must have put some sort of representation on the clustering.
So we didn't actually cluster. We used the clustering that was done by the Dick et al. Paper. So they, if I remember right, they used. Oh goodness, I don't remember what the PSLDA clustering with mathematic algorithm to cluster their single cell data set. So we didn't cluster anything ourselves. We just used, so when they, in their paper, they had a table that showed that in cluster 1, these are the top genes that represent cluster 1. And then we just took our RNA-seq data and matched it. So yeah, so we just did basically a manual clustering based on what they showed.
I have another question. I think it's a very general one also. Yeah. So because the post MI, the recovery is a big deal, for many patients, right? Are there any research effort that is trying to identify the possible biomarkers, like either chemokines or maybe the small molecules, which can indicate the recovery or anything you talk about here, right?
Right. Yeah. So I mean, that is something that we have done. When I was in my postdoc with Dr. Mary Lindsay, we had proteomics core where we were collecting plasma from post-MI patients, and we were doing proteomics on patients, looking at immediately after they came in, after their insult, and then after they were left the hospital, and trying to find out what possible biomarkers could correlate with the eventual development of heart failure, things like that. And so one thing that we did identify, and this we ended up focusing on sex differences, was that the lipid profile, again, we're gonna go back to lipids, but the LXR, RXR pathway seemed to be upregulated in men, downregulated in women, and that may be playing a role in that those play a role in heart failure.
Gotcha.
And so we have been looking at that historically. It's a little harder to do because mapping out those patients up until heart failure can take 5 to 10 years sometimes. And so trying to find out what markers are the best markers, I think a lot of people are doing. And we have a couple really good ones, and some of them, maybe not as accurate as others. But I mean, interleukin-6 tends to be one that people use. CRP is one, but that's not really specific for heart. So we do have a couple, but again, we tend to go right back to lipids every single time. And so again, I think that's why I tend to go that route, as I think there's really something there that maybe we need to really evaluate.
I think that's the Chris and me, we had this very strong interest in that field. Is can we use the data science to really help the to identify a better, or have a better way to predict such kind of.
Yeah. And so I mean, we were, and I don't know. I'm sure Mary's continuing that on now that I've left, but collecting plasma from multiple post-MI patients. So we were using big data sets like the Jackson Heart Study. You can use Framingham, things like that, to kind of try to identify possible biomarkers that will eventually develop, figure out which patients are going to go to heart failure.
Okay. It seems like even the sort of the analysis sort of the shifted time delay. That's true. I mean, in terms of the onset and the jump, but you still are missing a lot of the derivative. The increase is quite steep.
Yeah.
3 days. It's
Yeah.
much larger
Yeah.
impact than the other. So
Yeah.
if you aggregate, if you integrate over that point, you may actually have the same number.
Exactly. No, and
So you really, the sort of analysis of, after this point it jumps up, you forget the derivative.
Yeah.
You find it a lot. And
Yeah.
in fact, actually be incorporated more in the general scheme
yeah.
of the change in fact, which was just simply that onset.
Yeah.
Yeah, I had sort of more of a basic question about your conclusion about the collagen cross-linking. Is and you might have sort of mentioned this and I just missed it, but is that beneficial, or is that bad for the mouse or for the patient? I guess if you're talking about a human, that you don't have. What I was getting from it is that in your mutant mouse you had a more rigid collagen scar because there was literally more of it there, but it wasn't cross-linked. Is that?
So I will tell you that data is very conflicting in that it's more rigid, it's more stiffer, and yet it's less cross-linking. It doesn't quite make logical sense. Protein level, we go on with the lysyl oxidase, so I seriously doubt that that's going to be true if we look at all the different proteins that are important for cross-linking.
Okay.
So I don't think that that's true overall. I'm not positive that that is true. We do know that the scar is stiffer, and we do know that that is beneficial to a certain extent, because that's actually what helps keep that infarct from getting bigger. It keeps the LV from getting huge and dilating. So having that little bit of that structure is good. And so we want that. However, why it's stiffer, we still are trying, and it's Dr. Zion and Dr. Bradshaw and I had a meeting last week where we went back and forth trying to figure out, okay, why? Why is this happening? They told me, go back to your RNA-seq data, look at all the different enzymes. And I'm like, but none of them are different. So, we're trying to really figure out what is going on with the collagen as far as why is it stiffer, but we haven't quite teased that out yet.
So you haven't tried to measure the cross-linking biochemically, have you?
No, we have not.
Okay.
Yeah.
Any final questions from the students, Anybody? Let's thank our speaker one last time. You'll tell us what to do about it.
Oh, sure. So for the virtual audience, I'm going to end here. Thank you so much for attending, and we'll see you in January.